In this section, we will discuss genetic diseases of endocrinology and neurology. Let's start with endocrine. In order to understand these disorders, you have to understand the pathway of steroidogenesis, right? So we start with ACTH going to the adrenal glands, stimulating the development of the hormones from the cholesterol backbone that they're derived from. So we see the pathways here. We're not going to take time to go through all of these, as you will get them in other lectures. But the end result being the production of mineralocorticoids, glucocorticoids, and sex steroids, with the glucocorticoids being able to act in a negative feedback manner on ACTH to shut the system down when we have enough of all three of these things. So you can see various problems or various points at which you could have problems in the production of any one of these things. Particularly, there are diseases associated with the 11, the 21, and the 17 type hormones or type enzymes, and we'll get into that right now. So, a good way to try to remember hormone synthesis is to remember that we start with salt, then go to sugar, then go to sex. Or in other words, we start with the mineralocorticoids like aldosterone, then those derive into sugar steroids or the glucocorticoids, which then derive into the sex steroids, the androgens, and the estrogens. The production of mineralocorticoids and glucocorticoids involve 21-alpha hydroxylase and 11-beta hydroxylase. So deficiencies in these enzymes will give problems in producing aldosterone and producing cortisol. Then the production of glucocorticoids and sex steroids requires 17-alpha hydroxylase. So deficiency in this enzyme would cause problems there. And remember, this pathway is so interconnected that if you have one enzyme defective, all of the precursors will shunt down another pathway. So if you have 21-alpha hydroxylase deficiency, you're going to see salt wasting because there's no aldosterone being made, increased blood pressure, low potassium with 11-beta hydroxylase deficiency because of the involvement there as well. Now, if there's a 17-alpha hydroxylase deficiency, well, now we've cut it off and we're not able to go and move into the sex steroids. So what you see here in males, ambiguous genitalia because they're not getting the right testosterone production. In females, primary amenorrhea. Now, because of that 17-alpha hydroxylase deficiency, we have that absence of sex steroids. Let's go back and look again at 21-alpha hydroxylase and 11-beta hydroxylase. Again, if we don't have those enzymes, then we shunt all the way towards the sex steroids. So if we lack these enzymes, not only do we have the electrolyte imbalances that we discussed, but we're also going to see some different sexual characteristics. For example, males will get precocious puberty. Why? Increased testosterone production. It's shunting all of those precursors. And females will get ambiguous genitalia. Likewise, as we continue on with 17-alpha hydroxylase deficiency, you can see increased blood pressure, low potassium there as well. So looking at this on the diagrammatic or the schematic, we see if we cut out the 21, then we're cutting off the ability to make aldosterone, we're cutting off the ability to make cortisol, and everything gets shunted over to the sex steroids. So decreased mineralocorticoids, decreased glucocorticoids, increased sex steroids. And because we're not making that cortisol, we're not getting feedback to the ACTH. So we just keep getting more and more and more sex steroidogenesis, and that's what causes the things like precocious puberty or ambiguous genitalia in the females. Now, another point where we might see defects in testosterone signaling happens in the development of sexual characteristics. Now remember, in the embryo we have indifferent gonads that require stimulation by the SRY antigen to be formed into embryonic testes. From there we get Sertoli cell and Leydig cell differentiation. The Sertoli cells produce Mullerian inhibitory factor, which then cause Mullerian duct regression. Remember that we have to have this going through in order to stop the default female pathway, right? Now, in order to promote the Wolffian ducts, in order to promote male gonadization and sexual characteristics, we need testosterone. Testosterone can then stimulate the Wolffian ducts, which will then differentiate into the vas deferens, the seminal vesicles, and the epididymis, as well as turn into or be converted into DHT, which is important for development of the penis and scrotum. So, both of these steps require androgen receptor. What happens if you lack an androgen receptor? You make the testosterone, but you don't get the right characteristics. So these are XY karyotype females. Autosomal recessive deficient patients look like women, so we don't reassign sex here. They present with primary amenorrhea, blind vaginal pouch. We remove the remnant testes, and this condition is X-linked negative. So here again we see what's going on. If you lack the androgen receptor, then you will not develop male characteristics. However, you're making MIF because of that SRY antigen, so you will get Mullerian duct regression. So on the outside you will look female, on the inside you won't have the female internal organs. Another disease we see is with 5-alpha reductase problems. This is an XY person. Again, they'll have ambiguous genitalia or a small phallus with undescended testes. Why? Because 5-alpha reductase is required to convert testosterone to DHT, and without that you don't get proper formation of the penis and scrotum. Now let's talk about a few autosomal recessive neurological conditions. Metachromatic leukodystrophy is a deficiency of aryl sulfatase A, which participates in myelin catabolism. So what you see here is metachromatic urine granules for diagnosis. Krabs disease, or globoid leukodystrophy, is a defect in galactocerebroside beta-galactosidase. Patients have diffuse lack of myelin, causing rigidity, dysphagia, and deterioration, kind of like a multiple sclerosis type picture. Wilson's disease is hepatolenticular degeneration. This is an autosomal recessive defect in copper metabolism with toxic accumulation of copper in the liver and the brain. Some of the giveaways for this one on the boards, you're going to look for chorea, and you're going to look for Kaiser-Fleischer rings in the eye. They will also have increased urine copper due to spillover. The serum levels will be variable. Some of the dominant neurological diseases are Fragile X syndrome, Huntington's disease, and myotonic dystrophy. These are all triplet repeat type diseases, as we talked about in the past. So let's kind of go into depth as to what that means. Fragile X syndrome is a triplet repeat for the FMR1 gene. Huntington's is a triplet repeat within the Huntington gene. And myotonic dystrophy is a triplet repeat associated with the myotonin kinase. So with this triplet repeat disorders, we have something called anticipation. This is the worsening of disease severity or earlier manifestation of disease with subsequent generations. It's classic for triplet repeat mutations because these mutations are unstable and they expand during meiosis. So you'll see with each passing generation, the disease either occurs earlier and earlier in life or gets worse and worse. A good example of this is spinocerebellar ataxias. This disease is caused by triplet repeat expansion of the coding sequence leading to polyglutamine repeats. The gene is unstable during meiosis, leading to triplet repeat expansion, like I mentioned, and it worsens the effects of the mutation within each passing generation. So Huntington's disease has an incidence of 1 in 20,000 Caucasians, specifically Caucasians. The disease is found in a mutation in the Huntington gene, which is on chromosome 4. And again, triplet repeat disease that causes polyglutamine stretch of amino acids at the beginning of the Huntington protein. This polyglutamine region is thought to result in a neurotoxic protein. This stretch causes this protein to become neurotoxic. Onset of this disease, look for males 35-40 years of age, causes a jerking, uncontrollable movement of the limbs, trunk, and face, progressive loss of mental abilities, and the development of psychiatric problems as well. For Fragile X syndrome, what we see is mental retardation, characteristic facial features, and large testicles in males. This is caused by a triplet repeat expansion of the FMR1 gene promoter. So it does not cause a polyglutamine expansion in the protein like we see in, for example, Huntington's, but this is a repeat expansion in the promoter. So what happens is you get hypermethylation and suppression of transcription. The CGG triplet repeat mutation is only unstable in maternal meiosis. So this means that minimally affected or unaffected women may produce multiple affected sons, but men without the disease cannot pass the disease on to their children even if they carry the permutation, because it's involved in the maternal meiosis. Incidence for Fragile X is about 1 in every 1,000 males, with females variably affected depending on lionization pattern and other factors. Rett syndrome, common, found in girls. Symptomatic, what you see, mental retardation. Incidence is about 1 in 10,000. The gene that gets mutated here is MECP2, which is a methyl CpG binding protein. This gene is found on the X chromosome and causes disease when present in one copy. So this is a good example of an X-linked dominant disease. Boys who inherit the defective MECP2 allele on their maternal chromosome die in utero. It's incompatible with life in males. So that's why we only see this in females, and it's an X-linked dominant disease, Rett syndrome. That concludes this section of the lecture. Next, we will discuss muscular and connective tissue diseases.